Genetics is the study of heredity and variation. Mendel discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. The pea plants are convenient for genetic study because they have many distinct characters, or heritable features, such as flower color. Each character variant, such as purple or white flower color, is known as a trait. Mendel chose to track only those characters that were true breeding, meaning that those plants only produce offspring of the same variety when they self-pollinate. When Mendel crossed the true breeding white and purple flower pea plants, which are the parent generation, he discovered that all of the first generation hybrids were purple. Then when he crossed the F1 hybrids, he discovered a ratio of about 3:1 purple to white flowers in the F2 generation. Mendel proposed that this pattern of inheritance is influenced by heritable factors, which we now call genes. The definition of genes in molecular biology is the sequence of nucleotides that encode for either RNA or protein. Mendel developed a model to explain his observations. First, he proposed that alternate versions of genes, now known as alleles, account for variations in inherited characters. For example, the gene for flower color in pea plants exists in two versions, one for purple flowers, represented as capital P, and one for white flowers, represented as lowercase p. Each gene resides at a specific locus on a specific chromosome. Second, for each character, an organism inherits two alleles, one from each parent. The two alleles at a locus on a chromosome may be identical, as in the true breeding plants of Mendel's pea generation, which are referred to as homozygous. The two alleles could differ, as in the F1 hybrids, which are referred to as heterozygous. Third, if two alleles at a locus differ, then one allele, known as the dominant allele, determines the organism's appearance, and the other allele, known as the recessive allele, has no noticeable effect on appearance. In this case, all the F1 plants had purple flowers because the allele for that trait, represented by capital P, is dominant. Mendel proposed the law of segregation, which states that the two alleles for a heritable character separate during gamete formation, what we now know as meiosis, and end up in different gametes. Therefore, an egg or a sperm gets only one of the two alleles that are present in the somatic cells of an organism. The possible combinations of a sperm and egg can be shown using a Punnett square, a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. The horizontal row and the vertical column each represents the possible allele produced by each parent. As predicted by the Punnett square, the hybridization of pea generation produces all heterozygous offsprings in the F1 generation. And the hybridization of the F1 generation produces three types of genetic makeup. Homozygous dominant and heterozygous offsprings account for the purple flowers, whereas homozygous recessive offspring account for the white flowers. From this result, we can distinguish between an organism's phenotype or physical appearance, such as purple or white flower, and its genotype or genetic makeup, such as homozygous dominant, represented by two capital P's, heterozygous, represented by a capital letter P and a lowercase p, and homozygous recessive, represented by two lowercase p. Mendel derived the law of segregation by performing monohybrid cross, a cross between individuals that are heterozygous for one character. Mendel identified his second law of inheritance by performing dihybrid cross, a cross between individuals that are heterozygous for two characters, which can determine whether two characters are transmitted to offspring as a package or independently. For example, in the pea generation, a true breeding yellow round pea plant is crossed with a true breeding green wrinkle plant, resulting in the F1 generation with all yellow round pea plants. From this result, it is clear that yellow is the dominant allele for color, represented by capital Y, and green is the recessive allele for color, represented by the lowercase y. Round is the dominant allele for shape, represented by capital R, and wrinkled is the recessive allele for shape, represented by the lowercase r. If the two characters are inherited together, the possible gametes would be capital Y, capital R, and lowercase y, lowercase r. And the Punnett square would predict a phenotypic ratio of 3:1 in the F2 generation. If the two characters are inherited independently of each other, there will be four possible gametes, capital Y, capital R, capital Y, lowercase r, lowercase y, capital R, lowercase y, lowercase r. The Punnett square would predict a phenotypic ratio of 9:3 to 3:1 in the F2 generation. Since the phenotypic ratio of the F2 generation turns out to be around 9:3 to 3:1, Mendel proposed the law of independent assortment, which states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. In this case, the alleles for color, green or yellow, segregate independently of the alleles for shape, round or wrinkled, producing 4 types of gametes. The Mendelian laws can be linked back to meiosis or gamete formation. The law of independent assortment occurs during metaphase 1, where each pair of homologous chromosomes line up at the metaphase plate independently of other pairs of homologous chromosomes. The law of segregation occurs during anaphase 1, when pairs of homologous chromosomes physically separate from each other, resulting in haploid daughter cells by the end of meiosis 1. Strictly speaking, the law of independent assortment only applies to genes on different non-homologous chromosomes. Genes located near each other on the same chromosome tend to be inherited together, which will be talked about later in the video. Mendel's law of segregation and independent assortment reflect the rules of probability. The multiplication rule states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. The F2 generation can be predicted by the multiplication rule. Each gamete has 1/2 chance of carrying the dominant allele and 1/2 chance of carrying the recessive allele. Therefore, the probability of each genotype is 1/2 times 1/2 equals 1/4, as predicted by the Punnett square. The rule of addition states that the probability that any one of 2 or more mutually exclusive events will occur is calculated by adding together their individual probabilities. For example, to predict the probability of a dominant phenotype, one would add up the probability of the homozygous dominant and heterozygous, leading to the result of 3/4. The relationship between genotype and phenotype is rarely as simple as in the pea plant characters Mendel studied. Many heritable characters are not determined by only one gene with 2 alleles. The Mendelian genetics mainly applies to complete dominance, which occurs when phenotypes of the heterozygote and the dominant homozygote are identical. For example, a cross between a dominant red flower and a recessive white flower results in all red flowers in the F1 generation. In incomplete dominance, the phenotype of F1 hybrids is somewhere between the phenotypes of the 2 parental varieties. For example, red flower and white flower produces pink flowers. In codominance, the 2 dominant alleles affect the phenotype in separate, distinguishable ways. For example, red flower and white flower produces flowers that are both white and red. Many genes have multiple alleles. For example, the phenotypes for the ABO blood group in humans are determined by 3 alleles for the enzyme that attaches A or B carbohydrates to red blood cells. The enzyme encoded by the IA allele adds the A carbohydrate, whereas the enzyme encoded by the IB allele adds the B carbohydrate. The enzyme encoded by the lowercase I allele adds neither. Most genes have multiple phenotypic effects, a property called pleiotropy. For example, pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases, such as the sickle cell disease. Some traits are determined by 2 or more genes. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, in mice and many other mammals, coat color depends on 2 genes. One gene determines the pigment color, with alleles capital B for black and lowercase b for brown. The other gene determines whether the pigment will be deposited in the hair, with allele capital C for color and lowercase c for no color. Epistasis leads to phenotypic ratio of 9:3 to 4 in the F2 generation of a dihybrid cross, which is different from the 9:3 to 3:1 ratio of a typical Mendelian dihybrid cross. Many traits exhibit polygenic inheritance. For example, skin color in humans is determined by multiple genes. Polygenic characters typically have a broad norm of reaction, which is the phenotypic range of a genotype influenced by environment. The skin color is largely dependent on the environment as well. The first solid evidence associating a specific gene with a specific chromosome came from the embryologist Thomas Hunt Morgan, who discovered sex-linked genes through experiments with fruit flies. Morgan noted wild-type or normal phenotypes that were common in the fly populations, such as red eye indicated by W+. Traits alternative to the wild type, such as white eye, indicated by Y, are called mutant phenotypes. In one experiment, Morgan mated male flies with white eyes with female flies with red eyes. The F1 generation all had red eyes, but the F2 generation gets interesting. All the females had red eyes, whereas half the males had red eyes and the other half had white eyes. Morgan determined that the white eye mutant allele must be located on the X chromosome. Similar to humans, female flies are XX and males are XY. Each ovum contains an X chromosome, while a sperm may contain either the X or the Y chromosome. Following this hypothesis, the P generation crosses female with homozygous dominant red eye alleles with males with a recessive white eye allele, resulting in an F1 generation with heterozygous females and hemizygous dominant males, which all had red eye. Crossing the F1 generation results in all red eye females, with either homozygous dominant or heterozygous alleles. The males could be either red eye or white eye. Such gene that is located on either sex chromosome is called a sex-linked gene. In humans and other mammals, the SRY gene, which stands for a sex-determining region of Y chromosome, on the Y chromosome codes for a protein that directs the development of male anatomical features. In mammalian females, one of the two X chromosomes in each cell is randomly inactivated during embryonic development. The XIST gene, which stands for X inactive specific transcript gene, inactivates X by condensing it into a bar body. A calico cat results from a random inactivation of fur color genes in female cats. As mentioned earlier in the video, the Mendelian law of independent assortment only applies to genes located on different chromosomes. Genes located on the same chromosome tend to be inherited together. For example, genes A and B are both located on the same chromosome. If one homologous chromosome contains dominant A and dominant B alleles, while other homologous chromosome contains recessive A and recessive B alleles, meiosis will result in 4 possible gametes. Parental types follow the same genotypes as the parents, whereas recombinants have new combinations of alleles. If the inheritance follows the law of independent assortment, all 4 types of gametes have equal probability. However, if genes are located on the same chromosome, the probability of non-parental gametes depends on the frequency of recombination, abbreviated as RF. Recall that during prophase of meiosis I, homologous chromosomes pair up in the process called synapsis. Non-sister chromatids could exchange DNA segments at chiasmata, resulting in new combinations of alleles. Such phenomenon is known as genetic recombination, the production of offspring with combinations of traits differing from each parent. The frequency of recombination is the percentage of recombinants in total offspring. Higher chance of crossing over leads to higher frequency of recombination. More interestingly, the recombination frequency also correlates to the physical distance of genes on the same chromosome. The farther apart two genes are, the higher probability that crossover will occur between them, and therefore the higher the recombination frequency. Distances between genes can be expressed as map units. One map unit, or centimorgan, represents 1% recombination frequency. Map units indicate relative distance and order, not precise locations of genes. Recombination frequency has a maximum of 50%, since the recombinants cannot outnumber the parental types. 50% recombination frequency means equal chances of parental and recombinant types. 50% recombination frequency on the same chromosome is considered physically linked and genetically unlinked, basically following the law of independent assortment. Using the recombination frequency, a genetic map, or an ordered list of genetic loci along a particular chromosome can be constructed.